Hi everyone, welcome to the Division C Designer Genes live stream. I'm Stephanie and I'm joined by my friend Sophia.
Hi.
Hi. So just a quick about us. Hi, I'm Stephanie. I'm a current senior at Shadyside Academy, which is a school in Pennsylvania, and I've been in designer genes for the past 2 years.
Hi, I'm Sophia. I am a first-year student at the University of Virginia, and I'm a former captain of Syosset High School, which is in New York, and I've done this event for 2 years as well.
Cool.
So for a brief overview, in this event, you're allowed one sheet of information, two standalone non-programmable, non-graphing calculators, and the exam itself may be run as stations or a traditional written exam. So over on designer genes, what you want to look at is in what ways can genetic information be transmitted or passed down from parents to kids. So the basis for that is understand what a gene is. A gene is a sequence in nucleic acid, either DNA or RNA, to synthesize certain amino acid sequences. And the importance of that is understand genotype versus phenotype. Your genes will not always be expressed, and the phenotype is the physical traits manifested from your genes, whatever materials passed down. The allele is a variation of a gene. So you can have dominant and recessive alleles, where dominant alleles will overshadow recessive alleles, and a lot of genes will have a combination of two alleles. So you can have homozygous, where you have two of the same alleles, or heterozygous, two different alleles.
All right.
So in genetics questions for designer genes, you might be asked about different ways that traits can be passed down. Some traits depend on a combination of multiple alleles, like your human blood. You have type A, type B, type AB, type O. You also have some traits linked to your sex. So if you're male, where you have an X and Y chromosome, you're more likely to have those traits passed down. And there's also epistasis as well. There's also codominance and incomplete dominance. For example, with blood type and snapdragon flowers, which when you have, for example, an allele for white flowers with an allele for red flowers, the phenotype or the physical expression is a pink flower, a mixing. So that's called incomplete dominance. And then a lot of exams might ask about genetic disorders, how they're passed down, which could be autosomal, chromosomal, dominant, recessive. And since there's a lot of diseases or conditions they might ask about, we recommend that for these, you put them on your reference sheet. But you should be familiar with some really common ones like hemophilia or sickle cell anemia.
I'm just going to briefly talk about pedigree charts, human karyotype analysis, and blood types, because these most of the time show up on the tests, but are really brief. So this is a pedigree chart. It's of the royal family. They had a lot of inbreeding, so there was a lot of hemophilia that was passed down through generations, and basically like it's important to know like the genetic disorders and the inheritance patterns that Sophia mentioned in the slide before, to be able to like really identify and look through the chart to see what disorder like a family might have in their tree. And then this next diagram is of a human karyotype. And then this diagram to the right is just a diagram of like blood types. They can ask you what the blood types of like this circle number 1 and this square number 2 are, and you just have to figure it out. So basically next is biotechnology. And I just wanted to cover 2 subjects briefly, which are gel electrophoresis and PCR, that are pretty essential in understanding most of the biotech things that we have to cover in genes. So gel electrophoresis is basically a tool used to separate DNA fragments based on size. And it basically relies on DNA molecules having just like this innate negative charge where they're like filled into these wells at the top that are negatively charged, and at the bottom there's like a positively charged cathode. And as it runs, since DNA is negatively charged, it will move through the gel matrix towards the positive charge. And then smaller fragments will travel down farther to the bottom because they're just lighter and they can work through the gel more easily. And then larger, more like heavier fragments will be at the top. And this is just a great way to understand how you can separate DNA. And then to the right here is PCR, and basically PCR is, it stands for polymerase chain reaction, and it amplifies the amount of DNA that you have. So that's through 3 processes. I won't go into detail, but they're denaturation, annealing, and elongation. And basically it can take one double-stranded DNA piece and turn it into 2, and then 4, and then 8, and then 32, and so on until you have 1 million copies of this piece of DNA that can be used in testing. So in terms of biotechnology, there's just a lot of tech that can help us understand DNA samples, and the first ones are sequencing. So sequencing is used to determine the specific order that nucleotides in DNA are in. So Sanger sequencing is when a DNA fragment is denatured into a single strand, and then things like primers, polymerases, and nucleotides and like these fluorescent nucleotides are added to the mixture, and a machine raises the temperature so that this enzyme called the DNA polymerase can add nucleotides to that single-stranded template DNA and create a complementary strand. And basically, the process makes numerous strands, and they can be put into a gel electrophoresis and be separated by size. And eventually, once you've done this, you'll get a giant like data file that's put together on a colorful electropherogram that can basically tell you what the DNA looks like. And next generation Sanger sequencing is basically the same thing, only instead of like, since Sanger sequencing sequences a single DNA fragment, next generation Sanger sequencing is massively parallel and can sequence millions of fragments simultaneously in just one run. Next are microarrays, which is this colorful square to the right. It's basically an organized array of microscopic DNA spots on a slide, and they're basically used to simultaneously measure the expression levels of a large quantity of genes or genotypes in multiple regions of a genome. And they basically do this by using different fluorescent colors, like you see like red, green, yellow, orange, to display different levels of expression. Next is RFLP analysis, which is also known as restriction enzyme analysis. And basically a DNA sample is broken into pieces and are digested by restriction enzymes. And after this, these are separated using gel electrophoresis, and it can tell you a lot of things. And just in real life, for reference, it can be used for DNA fingerprinting. And then lastly, we have blots, and blots are basically used for identifying specific molecules such as DNA, RNA, proteins or anything in between in a mixture. And they basically extract the DNA, RNA, or proteins and separate them again through a gel electrophoresis. And then the RNA goes through an RNA membrane and is detected using specialized probes. And this chart here below are just the 4 different types of blotting.
In designer genes, in order to understand the biotechnologies that Stephanie mentioned as well, and to understand how you're going from your genotype to your phenotype, whatever genes you have, to the physical traits that are presented, you're going to want to understand enzymes. For example, DNA polymerase is often comes up on designer genes tests. You will want to know the functions of different types of DNA polymerase in DNA repair. DNA polymerase is responsible for creating a new strand in DNA replication through elongation. You'll also want to learn about restriction enzymes, which I think Stephanie will touch on a bit later. And like I said, so you want to understand how you're going from the genome to the phenotype. So going from DNA to proteins. So it's essential that you'll study going from DNA, which is split, and then you go to mRNA, and then that goes from transcription, translation, tRNA to the polypeptides, and then the proteins that will result in the traits being expressed. But not all the genes will just simply go from genome to phenotype. So there's types of gene regulation in both eukaryotes and prokaryotes, such as microRNA, which are small, non-coding RNA molecules that function in RNA silencing, and also post-transcriptional regulation of gene expression. So talking about the gene, you'll have to understand the structure of a gene in general. For example, here you can see the Tata box, which is a sequence of DNA found in the promoter. And the core promoter is found and marks the start site for transcription by binding RNA polymerases and other proteins that are needed to copy DNA to RNA. So some questions might be asked about like identify parts of the gene or given hypothetical situations involving if promoters are affected or other parts of the gene. So you'll have to understand what's happening with the gene as it goes from the gene to the protein and the parts of the gene. Another thing that comes up a lot in designer genes are operons. So an operon is a functioning unit of DNA that contains a bunch of genes under the control of a single promoter. So these genes are either transcribed together into one mRNA strand and translated in the cytoplasm, or they're spliced. So genes in an operon are either expressed together or not at all. One famous example of this is the lac operon. A harder question you might encounter on designer genes tests are if you affect certain genes. So for example, the lac operon, you have the 3 genes which translate for 3 proteins. These genes are LAC-Z, LAC-Y, and LAC-A. And the different proteins that they will translate to have different functions in cleaving lactose to glucose, transporting lactose into the cell, and so forth. So some tests will ask what happens if you like turn off a promoter or affect a certain gene, then what will happen in cells in this process.
Okay, so next we're going to move on to gene therapy, and really the super big part of designer genes is learning about this technology called CRISPR-Cas9. It's a gene editing tool, and this woman on the right basically helped create it. She won a Nobel Prize this year, and she's awesome. her name is Jennifer Doudna. So I'm just going to give you a general overview of what it is, but you should know that it's a lot more complex and there are a lot of steps and components that I won't explain that are just in the process, and you can research it a little bit more if you want to learn. So basically, CRISPR is a bacterial defense mechanism used to target and edit DNA in specific locations. And it's typically used for gene therapy and is currently being used to correct mutations that cause diseases. So it's um, pretty major in the field of medicine and biotechnology. And basically, like talking about the mechanics of it, it can basically be used to repair or study a gene which has a mutation. And in a gist, RNA sequences called cRNAs bind to a protein called Cas9, and not a protein, a complex called Cas9. And Cas9 can detect a complementary sequence in the DNA and basically cut the DNA and disable the gene. And then scientists can then introduce a gRNA into the cell so that the Cas9 targets a specific gene that they want to study the function of. And then also in designer genes, there isn't a lab section, but I thought that this is sort of what a lot of biology students do in the lab. And I considered a lab question on just cloning. So molecular cloning is inserting recombinant DNA into various host organisms and replicating them. And it's most often used to manufacture large quantities of desirable proteins. So, for example, everyone knows what insulin is. It's a hormone that's used in diabetes management. But basically, insulin is primarily produced using recombinant DNA inside bacteria, such as E. coli. Um, and moving on to plasmid cloning, if anyone doesn't know what a plasmid is, plasmids are just small DNA molecules located outside of the chromosome that can replicate independently. And plasmid cloning is used to transfer foreign genetic information, foreign genetic material into another cell. And they're typically used to clone and amplify expression of certain genes and can also be used in gene therapy, such as CRISPR Cas9. And you'll probably have to understand the processes it takes in a lab to do your molecular cloning or plasmid cloning. So listed here are just a few processes or important steps that you should probably know that will show up on a designer genes test. So here are just some resources that are pretty important. If you go on scioli.org and the designer genes wiki, there's so much more information in such greater detail of basically all the topics that we've covered. Sophia, do you want to talk about the textbook?
Okay, so Campbell Biology textbook is your standard textbook used in a lot of AP Bio classes. If you've ever studied for Olympiad, you'll know this textbook pretty well. This textbook will cover the fundamentals of, for example, you'll have to know the different types of genetic inheritance that you can study through this textbook, how DNA is regulated, the process of DNA replication, modification, different enzymes used in these processes. Operons are also in there. So like these fundamentals you can study and practice in most bio textbooks like Campbell. But for harder questions, like specifically modifying elements of an operon or like those biotech questions, using the test exchanges available and any competition test is a really great resource because that's the best way to know what type of questions you might see on tests that you'll take at competitions you attend. And specifically for CRISPR-Cas9, because it's more recent, a lot of pertinent information you'll find in science journal articles. Um, and if you're not sure how to access those or how to find those, uh, Googling them and then looking at the linked references, even on like the Wikipedia pages, you can find the relevant articles for that.
Cool. And if you have any questions, our emails are here. So feel free to, message us if there's something that you didn't understand or if we can do anything to help.